Hello everyone, this is Professor Anand Raghudan. So today we're going to discuss about the part number 2, okay, of the enzyme. So let's move it. So in this we're going to see the mechanism and the action of the enzyme. So catalysis is the prime function of the enzyme. Okay, we all know the enzymes are the catalytic, having the catalytic activity. So that is why catalysis is the prime function of the enzyme. The nature of the catalysis taking place in the biological system is similar to that non-biological catalysis. So whatever the chemical functioning of the enzymes or chemical reaction which are carried out inside the body, it similar having similar uh, chemical reaction uh, in non biological catalysis also. For the chemical reaction to occur, so for the any chemical reaction that occurs, the reactant have to be in an active state, or we can say the transition state. Okay, what exactly the transition state or active state? So it is defined as the state corresponding to the substance to the highest potential energy along with the reaction coordinate. Okay, then enzymes lower activation energy, which means the energy required by the reactant to undergo the reaction is known as activation energy. Okay, whatever the energy required for the undergoes the what we say the chemical reaction is, the whole energy is nothing but the transition state or transition energy we can say, or simply we called as enzyme activation, enzyme lower activation energy. So here is one example is given. If we see, there is one mountain. Okay, so if suppose we have to cross this mountain, we will go, what will happen, normal people will climb that mountain and will go from another side. So better option, we will make a tunnel between inside the mountain. So what will happen, it will directly reduce the energy. So this is what exactly enzyme function, it will reduce the energy of the chemical reaction and increase the rate of the reaction, right? So this is what the basic example of the chemical reaction. So the reactant when heated attain the activation energy, the catalyst, or we can say the enzyme, reduces the activation energy, and this causes the reaction to proceed at the lower temperature. Enzymes do not alter the equilibrium constant, they only enhance the velocity of the reaction. The role of catalyst or the enzyme is comparable with the tunnel made in the mountain to reduce the barrier as illustrated in the figure. The enzyme lower energy barrier of the reactant, thereby making the reaction faster. The enzyme reduces the activation energy of reactant in a such way that all the biological system occur at the body temperature, that is below 0 degree centigrade. Then enzyme substrate complex formation. The primary requisite for the enzyme catalyst is the substrate and combined with the enzyme at the active site of active site to form enzyme substrate complex, that is ES complex we can say, which ultimately result in the product formation. There are different theories, that is lock and key model, or we can say Fisher template theory. So in this theory, he was proposed by this theory actually proposed by the German biochemist, that is Emil Fischer, that is why it is called as Fisher template theory. According to this model, the structure or conformation of the enzyme is rigid. He said that enzyme structure and conformation is rigid. The substance fits to the binding site, now it is called as active site, just as a key fits in the proper lock or a hand into the proper glove. Thus the active site of an enzyme is a rigid and pre-shaped template where only specific substrate can bind. This model does not give any scope for flexible nature of enzyme, hence the model totally fails to explain many facts of enzymatic reaction, the most important being the effect of allostatic modulator. Okay, so this is what the first theory which is suggested by the Fischer in long time ago. Then second one is induced fit theory, or we can say the Koshland's model. Koshland in 1958 proposed a more acceptable and realistic model for enzyme substrate complex formation. As per the model, the active site is not rigid, okay, and pre-shaped. So as per the Fisher, he said the active site is rigid and has a pre-shaped, okay, but Koshland says active site are not rigid and are not pre-shaped. The essential feature of the substrate binding site are present at the nascent active site. The interaction of the substrate with the enzyme induces a fit conformation change in the enzyme, resulting in the formation of a strong substrate binding site. Further, due to the induced fit, the appropriate amino acid of the enzyme are repositioned to form active site of bring about the catalysis. Induced fit model has sufficient experimental evidence from X-ray diffraction study. Koshland model also explains the action of allostatic modulator and competitive inhibition on the enzymes. Then substrate strain theory. In this model, the substrate is strained due to the induced conformation change in the enzyme. It is also possible that when a substrate binds to the perform active site, the enzyme induces strain to the substrate. The strained substrate leads to formation of product. In fact, a combination of the induced fit model with the substrate strain is considered to be operative in the enzymatic action. So these are the different theories and mechanism of the enzymes. So we have discussed in these slides. Okay, thank you.